Hello and welcome everyone to GPS STEM Elevator Pitch Competition. This year, due to COVID-19 pandemic, we have taken the competition online. We have 9 PhD students from different STEM disciplines participate in this elevator pitch competition. I would like to thank our judges for taking time out to assess these elevator pitches. Before I give it to our coach, I would like to sincerely thank Bri McWhorter for organizing the elevator pitch workshop, one-on-one sessions with trainees, and organizing this elevator pitch competition. Thank you, Bri.
Hello, and welcome to the 5th annual GPS STEM Elevator Pitch Competition. Now, unlike other pitch competitions, where people are asked to talk about a particular research project or an entrepreneurial idea, this competition was created to help trainees prepare for the interview process. And in any interview, some form of this question inevitably gets asked. And that question is, tell me about yourself. Which is a very open-ended question because it gives you complete power to answer it in whatever way that you'd like. But instead of just going through a CV or talking about your academic history, instead, it's really an opportunity to share what you're passionate about why you do what you do, and what moves you forward. So we created this competition to help trainees prepare for that very specific event. And since most interviews have some online component, this year we have also gone virtual and asked students to submit videos of themselves answering that tell me about yourself question. So please enjoy and have fun learning about these wonderful individuals as they introduce themselves to you.
Hi, I'm Angelica Govea, and I love being a scientist. I love going into lab and doing translational research. I'm also proud of how far that I've come in my career as an underrepresented minority. As a Mexican-American woman, I did not encounter minority STEM role models while growing up. Instead, I discovered my passion for science in grade school, and from then on, I forged my own path to where I am today. During my studies, I discovered my interest in neural stem cell biology, molecular and cellular biology, and pharmacology. I also learned about the type of research that could be accomplished by merging these dynamic concepts, and I was hooked. Now I'm a PhD candidate at the University of California, Irvine, in the Department of Pharmaceutical Sciences. I'm currently looking at mechanisms underlying adolescent substance abuse. More specifically, my research is focusing on how a change in one DNA base pair and a non-coding part of a gene affects nicotine use in adolescents. Following the completion of a PhD, I want to become a principal investigator or a PI of my own independent research lab at a teaching university. I want to uncover the mechanisms underlying psychiatric diseases such as autism spectrum disorder. I hope to identify potential therapeutic targets and to better understand these neurological ailments. I also hope that as a PI, a principal investigator, I can be the role model and mentor for others that I lacked growing up. Visual representation matters, and I hope that by being seen in an academic setting, I am able to inspire others to pursue a career in STEM. Thank you.
Hi, my name is Rachael Lucas, and I think the human brain is fascinating. It's so complex and amazing. It's the most efficient computer in existence. And it keeps so many processes going at the same time, and in many ways keeps us alive without even a thought from us. But there are still so many unanswered questions about the brain. One interesting way to answer some of these fundamental questions would be to build a working brain model. Right now, no one is currently capable of this, but there are a lot of technologies that are working towards this. Currently, the closest thing we have to a human brain is a computer or a cell phone. Each of our smartphones contains 2 billion transistors. That's 2 billion moving parts working together to give us all the calling, texting, video watching, and game playing that we do on a daily basis. The human brain is even more complex, such that we're currently unable to build one. But we are able to work on some steps that are working towards this. And that's something that I get to work on in my lab. My lab works with nanopores, teeny tiny holes that can sense things and interact with their surroundings. These are made to mimic biological nanopores, which occur throughout all the cells in our bodies and play a huge role in our brains. Nanopores can act as building blocks so that we can use them to hopefully model a neuron, one single brain cell, and then by combining these together, more complex neural networks, until maybe one day a brain model. It's amazing to me to be working on technology that could be used towards getting us closer to understanding some of life's biggest mysteries, what makes humans human.
My name is Kelly Millar, and I'm a problem solver. One of the biggest problems in science today is understanding why certain people get chronic diseases. As a reproductive toxicologist, I ascribe to the theory of developmental origins of health and disease. This surmises that certain people are more prone to developing chronic diseases because of the metabolic makeup of the egg and the sperm that form the embryo from which the human arises. In my work, I seek to understand how air pollution can detrimentally impact the metabolism of female egg cells, or oocytes. To do this, I expose a pregnant female mouse to common compounds found in air pollution. This in turn exposes the female fetus and the oocytes are developing in that female fetus. Then, once the female is born, I retrieve the oocytes and compare various metabolic endpoints of the exposed oocytes to that of the unexposed. An individual's metabolism is predominantly determined by the oocyte. So within the context of developmental origins of health and disease, characterizing how something as common as air pollution can alter an oocyte's metabolism could provide crucial understanding as to why certain people get various metabolic diseases. Now, if this sounds niche, it's because it is. But that's because this is still a relatively new field with a lot that is not really too well known or understood. And that's what gets me so excited about this field. Because I'm used to solving really hard problems. From the time I was 3 years old, I was trained as a competitive figure skater. So I'm used to falling and failing. And when I do, I don't stay down for long. I get back up, I learn from what went wrong, and I try again. It's how I approach absolutely everything in my life. Especially my work, because when you're focused on something as important as reproductive toxicology, you need to get back up.
Hi, my name is Sasha Gatzke, and I am passionate about 3D printing. I bet you've seen the videos with cute little printers making iPhone cases, Eiffel Towers, and Baby Yodas. But 3D printing can go far beyond making little toys. It can help areas affected by natural disasters and epidemics. Right now, the supply chains all over the world are ravaged by the novel coronavirus. Many patients affected by the virus are struggling to breathe and need a ventilator. The valves for the ventilators are now in a dangerously short supply. An Italian engineering company stepped up and used 3D printing to make the valves. At just a few dollars a piece, these valves are saving lives today. 3D printing is an amazing technology that has enormous potential. From toys to medical devices to rocket engines, we can build almost anything. But some challenges still remain. 3D printed parts often have lower quality than parts made by traditional methods. This is crucial for aerospace parts where quality and reliability are paramount. Companies like Boeing, SpaceX, and GE would love to embrace 3D printing, but they simply cannot risk putting 3D printed parts into their engines just yet. To print a rocket engine, we need to switch from plastics to metals, but 3D printed metal parts often have lots of defects. They can warp, crack, and lose their shape. All of these defects make 3D printed parts weaker and more fragile, and can even cause catastrophic failure. I am working on taking metal 3D printing to the next level. I study the defects in 3D printed parts and come up with ways to avoid them. I achieve this by designing completely new metal alloys. These alloys are inherently resistant to the defects that plague metal 3D printing. I'm hoping that my alloys will help make 3D printed parts stronger and more reliable, so we could finally go from plastic baby Yodas to printing the Millennium Falcon. Thank you.
I'm Kristin Gabriel, and I care about making an impact. I'm so thankful to have joined the Weiss Lab, where we invent tools for chemical biology that can impact healthcare. DNA sequencing is a tool that we can use to unlock the makeup of DNA. It provides us with so much potential. For example, we're battling the coronavirus or COVID-19 pandemic. We need to fight the spread of this virus and the severe respiratory illness that it can cause. Knowing a virus's DNA sequence can help us make vaccines, and knowing the DNA sequence of patients can help us personalize medicine. In order to slow the spread of COVID-19 disease, we need faster and cheaper DNA sequencing. Currently, it takes a week and costs about $1,000 to sequence the DNA of a patient. Now, our lab invented a DNA sequencing technology that can read and record DNA in real time. Now, my role involves engineering the molecule that reads the DNA and discovering new ways to attach this molecule onto our sequencing instrument. Once we have the DNA, we can find differences and understand and unlock the DNA's information. I collaborate with the world leader in DNA sequencing, and our goal is to make DNA sequencing 10 times faster and cost 10 times cheaper so that we can make DNA sequencing become a standard in healthcare.
Hello, my name is Devashish. I'm a PhD student in the Material Science and Engineering Department, and I am passionate about renewable energy. We all use fossil fuels in our everyday life, right? From driving your car to the electricity that comes to your house and to the food that you cook on your stove. But we all know that burning fossil fuels releases greenhouse gases which cause pollution, which in turn leads to global warming, ocean acidification, and all other related phenomena. Therefore, we want to switch to cleaner energy technologies, and fuel cells are the best alternative. Fuel cells are electrochemical devices which convert hydrogen and oxygen into electricity. The only byproduct of a fuel cell is water, which is very safe and which can be reused. For us to switch from a fossil fuel economy to a hydrogen economy, we want to make fuel cells better, more efficient, and cheaper. The key to improving a fuel cell is to make better materials which make up the fuel cell itself. But the question is, how do you visualize the interactions, the material interactions, while the fuel cell is operating? Well, I use an imaging technique called X-ray computed tomography, which helps me visualize these interactions in 3D while the fuel cell is operating. These images give me valuable information in terms of the catalyst behavior, the gas transport, and the water formation in the fuel cell. By using this information, we can then engineer better materials with improved properties which can then make a fuel cell better and efficient. I think with the development of such cutting-edge technology, it's equally important to have a better leadership to drive forward these changes. Hence, I'm involved with many other personal and professional development activities, especially outreach activities which help to boost the STEM potential of high school students. I think with the combination of a better leadership, the cutting-edge technology, and responsible citizens, we can drive our society towards a cleaner energy future. Thank you.
Hi, I'm Brenna Biggs. I've noticed recently that we enjoy having boundaries. We draw lines to separate counties. We create borders to make states and even countries. We build homes to live in, cars to drive in. We enjoy having spaces to call our own. This makes us feel safe and like everything is in order. But you know what doesn't care about our boundaries? The air. Harmful air can easily come into our safe spaces. It gets everywhere. This makes air quality a global issue and one that I find fascinating. I'm interested in how we try to contain toxic air so that it doesn't get into our precious personal spaces. As a California native, I'm particularly interested in cleaning up the air in the Central Valley. If you've ever gone for a drive up the 99 freeway, you've probably witnessed a very fragrant and recognizable smell. Dairy farms. The cows and their manure don't care that we feel safe in our cars, and neither does the harmful air that they bubble and burp out. This air somehow not only penetrates in through our car windows, but it can get into our homes and our bodies as well. It can alter places where we live, work, and play. I want to contain nasty air so that our spaces and boundaries remain safe and clean. On a current project, for example, I'm helping dairy farm operators reduce emissions in the Central Valley. With my help, they'll be installing new infrastructure to cover and trap harmful air and convert it into electricity. By being a part of projects like this, I hope to not only power, but also empower local communities to make this a greener and cleaner place to live. That way the world can breathe a little easier. Thank you.
Hello, my name is Veda, and I'm a PhD candidate at UCF. I strongly believe that just because something hasn't been done before doesn't mean it's not worth exploring. This belief is encompassed in my projects, whether it be in research or in outreach. In my research, I study particle physics. Specifically, I try to expand upon the theoretical model of the universe. Currently, the standard model in the field can only explain how matter behaves. Matter makes up all the stuff you've ever learned about, whether it be the moon, the stars, your food, your pets, and even you. However, matter only makes up about 5% of our universe. That means that our current theories and models can only predict how 5% of the universe will behave. Another 25% is a different type of matter called dark matter. This is what I'm interested in. Dark matter cannot be seen, and hence its name dark. It can only be observed through gravitational effects. There is no viable theory that connects how dark matter, and gravity all work together to create the universe that we live in. My belief is that my research can not only improve upon these models, but can also add to society in a different way, mainly by inspiring other women of color to join careers in STEM. Unfortunately, many women of color, myself included, have not had enough people in positions of power encouraging us. I hope to change that. I look forward for the opportunity to become an academic advisor or a professor to be able to give the necessary mentorship and support for women to thrive, not only in STEM, but in life. Because after all, in research or in outreach, it's up to us to look for opportunities where we can improve and add to society, even if those opportunities aren't always straightforward or easy. Thank you so much. I hope you've enjoyed my talk, and I hope that I cannot take you out of your apartments for at least 2 minutes with this background behind me. Have a great day.